Good morning, and thank you all for being here today. I'm going to present something very different. We're not doing AAV, we're a non-replicative herpes vector platform, and I will go into great detail about what this means. We're a company, we're based in Paris. We have our clinical development activity here in the US, and we've raised at this stage about $50 million. So what do we do? Our aim with our platform is to go and treat large patient population with chronic disease in the neurology field, be it the PNS or the CNS. And we believe that we can do this treatment of large patient population because of the inherent qualities of non-replicative herpes vector. We have a first product which is in clinical trial right now in the US, and I'll be speaking mostly about this, in fact, this morning. Second product which reached proof of concept in animal model and are developing a broad pipeline behind this. This is the result of a whole team effort. One person I'm going to mention because she's also relevant to the rest of the discussion is our Chief Medical Officer, Cornelia Hagmolken-Teller. She's the lady who, in the field of bladder disease, have brought 3 of the last drugs to the market for clinical development, including Botox, which will be the analogy here today. So why are non-replicative herpes vectors such interesting tool? It's a large vector. You can put a lot of genetic material in it. It has 2 tropism, epithelial cell and neuron. It's a vector that is very well tolerated. It doesn't generate any strong immune reaction. You can express gene in it for a very long period of time. It's cheap to manufacture with comparison to the AAV. And for us, the major property that we're using is the fact that when you inject it at a specific site, it then retrogrades itself through the nerve ending that are at that site to the nucleus of those neurons, and hence you can influence the activity of those neurons on a very local basis, which is what we do. So with that, we're focusing our effort on 3 different activities. The most advanced one is inhibition of type C sensory neuron, and I will cover this more in detail. So this first product, EG110A, has been built to inhibit the activity of type C sensory neuron, and here we are looking at it principally in disease that affect the bladder. So how is it designed? It's a vector that is designed to inhibit CGRP-positive neuron, of which type C sensory neuron are mostly CGRP-positive neuron. We use as a transgene an enzyme that degrades a protein called VAM2. If you degrade VAM2, you don't get the vesicle to bind to the synapse membrane, and hence you interrupt the activity of those neurons. And we put this transgene under the control of the CGRP promoter so that we get specific activation of it only in CGRP-positive cell, again back to the type C sensory neuron. The way your bladder works is a normal bladder function is driven by A delta sensory neuron. The abnormal reflex pattern that occurs in some disease setting is driven by type C sensory neuron, hence the tool that we've designed here with EG110A. In the current market, if you look at the various indications with those type of bladder pathology being neurogenic bladder, overactive bladder, or interstitial cystitis, in the 2 first, the major drug that is being used is Botox. The Botox is a great drug. The problem is its side effect. Botox does in your bladder what it does on forehead. It paralyzes the muscle, so in 30% of the patient, the clinical trial that they did, it generated acute urinary retention. That has prevented Botox from penetrating large segment of the market. We don't act in the same way, and hence we should not see the same type of problematic. Interstitial cystitis, which is a third indication, is of a different nature. There at this stage there's absolutely no treatment. So those are the 3 indications we're focusing on with this first product. So how does it work? For the practicing urologist will be the one dispensing the drug. For him, there's no chance versus Botox. Same equipment, injection in the bladder wall, 30 points of injection, and there starts the differences. Our product then finds the nerve ending, retrograde itself to the dorsal root ganglia, to the neuronal body, and then express itself in the nucleus of those cells to block their ability to transmit information downstream to the motor neuron. Botox, to the contrary, acts at the interface between the motor neuron and the bladder muscle to paralyze basically this tissue. So with us, what we expect decrease in urinary incontinence episode. We do not impair the normal voiding function, and in fact, all our preclinical work did show that we did not impair it. We should not increase residual volume of urine, which is one of the problems that Botox has also. And because of the very nature of our vector, it should last for a very long period of time, at least several years, if not longer. So from a market perspective, with that type of target profile, we should be able to get very large market expansion. As you can see, the current treated patient population with Botox across its both indication is basically 750,000 patients. And as you can see in the other bubble, we should be able to expand that very significantly. So the exciting part of what I have to share today are initial clinical results. Here, the first indication we're studying is neurogenic bladder in spinal cord injury patient. You find neurogenic bladder in spinal cord injury patient, in MS patient, in Parkinson, and so on, in anybody who has a neurodegenerative disease of the central nervous system, which interrupts the proper communication between the CNS and the bladder in the spinal cord. So our first target is the neurogenic bladder in spinal cord injury patient, because that's a very clean patient population from a profile perspective. The disease is set, it doesn't change, they have low placebo effect. So we started this phase 1b study in 4 centers in the US, and after that we'll do a phase 2b/3 study to get to a broad labeling. The FDA granted us last week fast track for our product based on some very early result that I will share today. So this study was a dose escalation study, 3 dose, low dose, mid dose, high dose. Low dose was 110 to the 8 PFU. The main outcome variable here are from a safety, of course. We also look at reduction of urinary incontinence episode, quality of life, and urodynamic variable at 3 months. We had our first cohort of patients was 4 patients. They were coming from 3 different sites, so 3 different independent investigators. The mean age was standard in this patient population, so they were not different than any population of SCI patient, even though the number is small. And out of the 4 patients, we had 1 woman who had a neurostimulation device that was not working for her. So what did we see on the adverse event side of thing? We didn't see any serious adverse event, and the only adverse event we saw were around the time of injection in 2 of the 4 patients, mild fever with nausea. It's worth stating here that none of the patient is pre-treated with any steroid before treatment, which is in contrast with AAV type of approaches. So this is, and then from an efficacy perspective, those are the results that we had, which made us feel great about the progress that we were making, because as you can see, the average here is at baseline, patient had 26 episodes of incontinence during the week. At 5 weeks, they were down to 5, and it got slightly better at week 8 and at week 12, so that basically we had almost a 90% reduction on urinary incontinence episode by month 3, which basically means it's basically maximum efficacy that you could expect to see in this type of setting. So those results were for us very important. Opinion leader that we spoke to validated that indeed those results were exciting, and they have put us in a place where we got the fast track with the FDA, and in fact are changing the way we're doing the rest of the study, because we're going to do the medium dose. We're not going to do the high dose, We've already enrolled 2 patients in the medium dose. We don't expect significant difference from an efficacy standpoint given where we are right now. And we'll address what we do with the patient that we're going to be in the 3rd dose later on. So with all of this, 3 months, the urodynamic measurement also moved all in the right direction, be it maximum cystometric capacity or the volume of urine at first void, as well as the overall compliance of the bladder, which compliance is a metrics that measure the change in volume of the bladder per increase in pressure. So you want high compliance because it shows that you're able to put a lot more urine in the bladder for less increase in pressure. All of this is moving in the right direction, and the quality of life questionnaire on incontinence is also showing great progress. If you contrast that to what Botox has published through their phase 3 studies, if you look at it, the baseline characteristic of the patient population in terms of numbers of episodes was not fundamentally different. It's 28 versus our 26 something. But as you can see, at different periods of time, the most that Botox achieved is a bit more than 50% reduction in incontinence episode. So with this, we believe that we have, on a small number of patients, compelling evidence of efficacy with a really good side effect profile, and we are, as I said, enrolling our 2nd cohort. We expect to share more data by some point during the month of November. So with this, that has also made us move our therapeutic product profile, our TPP. We expect now to have a product that has superior efficacy to Botox, no increase in the post-void residual volume of urine in the bladder of the patient, which is important for AB and MS patient in particular, and last a long period of time. At this stage, we have data up to 3 months. By December, we'll be at 6 months. To have a truly superior product, we believe that we need to be beyond 6 months of efficacy, and then we have something that is very strong from a profile perspective. The next product we have is EG-1428. This one does something very different. It codes for an enzyme GAD67 that transforms glutamate into GABA. So for us, it's the opportunity at the very local level also to modify the balance between excitatory and inhibitory function, and hence have an effect in a series of pain disease such as neuropathic pain. In fact, the first evidence of efficacy we got in a preclinical model is we demonstrated in a neuropathic pain model that indeed our approach worked as well as pregabalin, which is one of the drugs that is used to increase GABA level in a series of those type of disease. So we're doing more preclinical study there across different indications also, and the goal is to bring this product to an IND by some point in 2028. Beyond that, we're building our entire platform, and I'm going to this platform. I think the best way to describe it is to show an example of the type of complex structure that we can build in a herpes vector because of the size that we have and the different element of control. We build one vector that expresses one gene under short-term control and one gene under long-term expression control, so that we can get one gene only there for a very short period of time and then another gene for a much longer period of time. And as you can see on the right side, the expected behavior is to get the M green lantern expressed for a long period of time, while M scarlet decrease very quickly in this proof of concept side of thing. I realize I'm missing one slide here. The slide that is not there that you would have seen is the slide that shows that in fact in vitro and in a rat model, in fact, we obtain exactly that type of pattern with short-term expression of M scarlet disappearing very quickly. Since then, we've done another experiment which did work, because obviously one of the applications of this here is in the gene editing field. And we've demonstrated, and this is an in vitro experiment only at this stage, that by putting a CRISPR-Cas system in the short-term expression, we were getting indeed appropriate gene editing done while we were keeping the long-term expression of the other transgene going. So thank you very much for your time. I think we believe that non-replicative herpes vector have a great potential in front of them across a broad series of disease. Crystal Bio has already shown that on the epithelial side of things, and we hope to be able to show that in the neurology field. Thank you very much for your time.
Thank you, EG 427.